So without further ado, let me introduce Professor Kevin Shakesheff, who's Professor of Tissue Engineering at the University of Nottingham. He's also, for his sins, CSO of Regenmetech, and is going to take us through some of the opportunities he sees in transforming therapies. So over to Kevin.
Thank you very much, Kevin, and thanks very much to the organisers for the great organisation of the event and for giving me the opportunity to speak today. What I thought I'd do, because I guess it's a fairly diverse audience across life sciences and biotech, is give you sort of an overview of where I think we are in stem cells, tissue engineering and regenerative medicine at the moment, and then drill down to a bit of detail in my own research area, just to give you an idea of some of the challenges that many people can get involved, because the field is so multidisciplinary, and increasingly as we learn more about the challenges of getting some of these very complex technologies through to the market and hopefully through to the patient, we're learning that we need more and more people to get involved, and I'm sure there's roles for many people here today. I am slightly nervous from the point of view that my talk managed to crash the IT system about 5 minutes before we started, so one or two of the slides might not run as smoothly as possible, but I'll try to improvise around those when it happens. So I think the first thing to say is, as I'm sure people are aware, regenerative medicine creates so much excitement because we are looking at restoring functional tissue, and in comparison to some drugs, in comparison to many biomaterials, we're not just patching over the problem, we're actually looking at returning the patient to a fully healthy state, which, depending on the disease state, may stay with them for the rest of their lives. So these MRI images just give me a prompt to give you some ideas of the areas that people are looking at to do on this. So we've got on the left-hand side the human heart, to remind us that after an infarction or heart attack, the loss of tissue and the loss of tissue function is not reversed. So people have to live perhaps with drug therapy with a suboptimal heart, and that has an effect both in terms of quality life and possibly on length of life. So if a therapy could actually return that patient to having a fully functional muscle within the heart, then it would be a radical breakthrough. The second movie playing is looking at the human brain, and here in fact Scotland is playing a world-leading role with the first stem cell trial being conducted, I think, in Edinburgh, looking at injection of cells into the brain in stroke therapy. That's, I think, the first phase 1 clinical trial of its type, certainly in Europe. And again, it offers us potentially a route to therapy in an area that is just not well treated by drugs. Perhaps slightly more mundanely, but I think very importantly, on the right-hand side, we look at the skeletal muscle system and our joints, which were not designed to allow us to run around for 80 years. Again, the therapies for those are quite often implants, which, while they've been optimized and are extremely impressive, do not return you back to full health. And the area of regenerative medicine is making great strides in this area to take either the bone or the cartilage tissue and reform a fully functional structure that can stay with that patient for many years. So that's the vision, and it needs to be big because the problems of achieving that are massive, and we really need to be sure that if we put all this hard work over what I'm sure is still going to be many decades, that what comes out of it is a massive breakthrough for human health. Looking across the human body, I think it's fair to say that every single tissue of the human body is currently being studied from the point of view of can we regenerate healthy tissue. And the UK can be really proud of its position in many of these areas. I won't go through all of them, but I just wanted to make a general point that when you look at regenerative medicine, you'll see two types of tissue that we're trying to grow in general. Those where the adult can already regenerate it, so the biology is helpful. So if you take bone repair, if you take skin repair or liver regeneration, there's underlying mechanisms in all of us within this room that will try to regenerate those tissues. Now, something may have gone wrong due to a disease or due to a major injury, so that it's not currently able to regenerate. But at least if we can make an intervention and kickstart the system again, the biology is helping us, and we should hopefully see most rapid progress in those areas. There are other tissues which none of us in this room have an inherent ability to regenerate. So cardiac muscle, spinal cord, nerves, islets in diabetes, lung tissue. And here the challenge is greater, and from a scientific point of view, this is perhaps the most exciting areas, because there we need to look back to developmental biology and say, can we create environments which restore what happened in the embryo when these tissues were able to spontaneously form? And can our therapy actually capture what happened in development? So the challenge, and certainly Scotland has played a major role in this, is that the therapies are going to be very complex. We were talking before with Kevin about his experience in biopharmaceuticals, proteins, DNA. In their own right, a major step forward for the pharmaceutical industry, but also huge complexity going from small molecules to biological molecules. What regenerative medicine needs us to do is now to think of these things as the product. Living cells, difficult to characterize exactly what they are, very easy to make them not do what they're meant to do, but how do we ensure quality at the point of therapy of something that has so many different functions? And from an industry point of view, I'm sure Malcolm's thinking of 1,000,001 problems of how you can actually get this through. From an academic perspective, there are some fantastic new challenges for us to get our teeth into. This slide is just to sort of remind us again as we go through this that the biology can be very helpful. And in this particular example, we're looking at a movie of peripheral nerve regeneration. And the nerves have grown across from left to right over about a 24-hour period that we've sped up to a few seconds. And it's quite a complex process of the yellow cells that you can see are actually Schwann cells, which are the support cell structures. And they're providing the environment for the neurites, which are the very fine grey structures, to explore space. And this is really just us seeing a natural process which allows patients to regenerate peripheral nerves. In other circumstances, we're not so lucky. So in some of the areas in cardiac repair, in central nerves, this inherent ability of the cells has been lost, and we're going to have to work harder to get it back. Most of the examples I'm going to give you today are in the orthopaedic area, which is an interesting one for regenerative medicine. Again, I was speaking with our chair just before the conference started about whether regenerative medicine should go for the really big win, so regeneration of spinal cords, curing type 1 diabetes, or whether it should focus on low-hanging fruit and pick off some of the easier challenges. We're coming at it, and my work is coming at it in the second way, which is to look at orthopaedics as a very attractive first target, perhaps not the biggest financial win, but still plenty of opportunities, because the current surgical procedures are perhaps surprisingly a long way off optimal. So in orthopaedics, we're talking about regeneration of bone in this sort of scenario that you see in the X-ray on the bottom right-hand side of the image. Oh yeah, sure. Yeah. Okay. 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 Thank you. And so what we're looking at this situation in this patient is a major break of the bone, in which the spontaneous regeneration that we all should be able to do with bone tissue is lost just because of the scale of the injury. And there's plenty of this that occurs, and the market is great enough. The nice thing with bone is that the judging success is relatively straightforward, because you can look at mineralization using a non-invasive X-ray. You can judge how well that patient is doing very easily, and normally within about 3 months of making the intervention. So thinking about how we're going to judge success, there are simple markers to do that, and it's going to happen within a reasonably short period of time. The current approaches surgically are to either take some healthy bone from one part of the patient's body and to move it into this damaged area, which works well. The patient's own bone is a very good template for new bone formation. But of course it means that the surgeon is damaging their own patient, and quite often they get complications from the donor site which are worse than what you see from the... is that okay now? Suddenly come on. Can you hear me okay at the back? Is that okay? So what you get then is more injury being caused by the surgeon themselves at during the operation than what they're actually trying to solve. The other alternative is this allograft option, which is where you take cadaver bone, shown in the top right, just some femoral heads there. Lots of cost involved, actually, that's quite buried within the NHS in just processing this tissue, ensuring it's disease-free, and a shortage at times of that tissue. So we'd like to create some simpler solutions where we can get bone to form rapidly without some of the current problems of the current surgical procedures. Could you just advance the slide? I'm not sure it's... Okay, so just a little bit of an advert for Nottingham, just in terms of our role, certainly within the UK in this area. About 6 years ago, we set up with Wolfson funding the Wolfson Centre for Stem Cells, Tissue Engineering and Modelling. We've been fortunate to become part of the EPSRC Centre for Innovative Manufacturing in Regenerative Medicine that started last year. And we also have a major programme of training 50 new PhD students. What I think we bring to the party within the UK that's different to all of the other university groups is a mix of people who've come from a pharmaceutical background with stem cell scientists, material scientists and clinicians. And it's really that awareness of what's worked well in the pharmaceutical industry and can we learn from that when we're thinking of these complex stem cell therapies that I think hopefully are our unique contribution to the field. Thank you. Just to give you an overview of what the industrial opportunities are, and then to look at some of the challenges on the next slide. I like to characterise the products that are going to come out and that are coming out into 3 categories. The first is drugs that stimulate tissue repair, sometimes neglected in the excitement for cell therapies, but there has been considerable success in looking at molecules, mainly proteins, that if you put them in the right place within the body, will spontaneously allow tissue to regenerate. Probably the most successful of these is the bone morphogenetic proteins, which Medtronic have turned into a successful product in spinal fusion procedures. There's also a lot of interest in small drug molecules and looking at stem cell biology and what it can tell us about new targets for hopefully small molecules which will cause regeneration to occur. The second category then, and the one that gets the most attention, is the concept of a cell being a product. And there's a whole range of ways of approaching this problem. The simplest and the one that's reached the clinic most rapidly is autologous treatments. So for example, in cartilage repair, there's this process called autologous chondrocyte implantation, where you take healthy cartilage from the patient, say from their left knee, and you transfer those cells to the right knee that's damaged, with an intermediate step of expanding those cells under GMP conditions in a laboratory. So you're moving cells from the same patient, you're changing sites, and you're going through an expansion step in the meantime. There's then allogeneic sources. So Reneuron starting this trial at Edinburgh of an allogeneic source, which is from a fetal source that's been temporarily immortalised for stroke treatment. This has the advantage that you have just one product, and you can quality control that, expand it, and that can hopefully be used in many, many patients. And then we move on to the pluripotent stem cells. Here we're looking at turning the clock back, as I say, to the developmental stage, looking at cells that were able to form all of the tissues of the body when we were an embryo. And indeed, there are trials now just due to start or have started recently in the US, looking at spinal cord regeneration using embryonic stem cells, and not too far down the road, the concept of inducing pluripotency in adult cells. So the third product opportunity, and the one that I mainly focus on, is the concept of using biomaterials to stimulate tissue repair. And as I'll describe, actually the materials offer you a route of binding together the drug and the cell opportunities to create multifunctional products. Lots of challenges for the industry, and therefore lots of opportunities for innovation to overcome these challenges. I'll be here the rest of the day and this evening, very happy to debate these with people. Just a few. First of all, defining what is the product. If you have a living cell, how, from the regulatory point of view, are you going to write your specification? Is it to define the cell precisely? Is it to define one feature of the cell that must be functional? Or do you define it by the manufacturing process and just say if we make the same process happen time again and again, we'll end up with the same product? So that's a major issue. Attrition is a huge issue. The UK industry, and I think it's true across Europe, less so in the States, is really not set up for the high attrition rate that we're bound to see in these types of therapies. So there are too many companies out there that are single technology that really can't afford a single bad result. And I contrast that with some of the companies in the States that have got multiple platforms and are able to survive what's inevitably going to happen, which is some of the therapies are not going to work well in the very first trials. Clinical trials and health economics, this is a big area because in many cases the effect that we want to see clinically has to be seen for a long period of time. So the trial has to be very long. And in general, these are expensive interventions. The therapy is going to be very expensive. So from a health economic point of view, we're going to need to prove that the benefit is long-lasting and has large efficacy. Other issues around manufacturing and cost of goods, complex manufacturing processes, how do we deliver those at a cost that health services can afford? Mechanisms of action are not fully understood, so many of the therapies that are going forward, we're not sure whether they're protecting the stem cells that are already there, creating new blood vessels, or actually participating in new tissue formation. And there's the age-old issue of the role of the patent within this field, and patent lifetimes not being long enough for the current generation of cell therapies, which have taken perhaps longer than we'd expected to get through to the market. Just very briefly, in my own area, we see innovative material science as being one of the keys to solving some of those challenges on the previous pages. So thinking, as the pharmaceutical industry has had to think for many years, about the role of the formulation, not just the drug, but how are you going to deliver this into the body? Looking at local mechanical properties, so if you're looking at delivering into heart tissue, into the brain, or into bone, the mechanics of those tissues is very different. And if you don't match your delivery system's mechanics to the local tissue, you're going to cause a lot of scar formation, and you're probably going to impede the tissue from forming. Some quite mundane but really important issues about just how do you transport the product. If you're going to cryopreserve it, there are all sorts of issues around how you prove that process works and that you remove the cryopreservant. If you're going to transport live cells, how do you physically do that at a cost and with quality guaranteed in the process? And also in terms of intellectual property, we see some of the strongest IP within the cell therapy field, not being the cell itself, but in being the delivery route. Because there's so much controversy around intellectual property of cell therapies, the IP is dating, is that new innovative delivery systems might offer the type of protection that investors and companies need to invest in clinical trials. So this is our approach towards the field, and it focuses on the left-hand side on these products which are called scaffolds, which are materials. They come in many shapes and sizes. They're made of many different types of chemistries, but they all share the ability to host stem cells and to create a three-dimensional environment in which those cells will form functional tissue and will get the blood systems growing in that they require. We use scaffolds in three ways. The first is just as a system in its own right that if you put it into the patient will create an environment where the patient's own stem cells can migrate into that scaffold and will form tissue. That's the simplest solution. Unfortunately, it's fairly rare to find a clinical situation where that works. The one key example is in bone repair, where there are successful products now on the market, where just putting the scaffold into the damaged bone creates the right architecture, the right biomechanics for neighbouring stem cells to move in from within the patient, repopulate that area and form new tissue. In most examples, we're going to need more of a stimulus for tissue to form. And here what we try to do is still keep the scaffold as the platform, but introduce one or two functionalities. And going up the top pathway, one opportunity is to use the scaffold as a controlled drug delivery system for these growth factors or small drug molecules that will cause new tissue to form. So there we go into the pharmaceutical realm, the very successful area of controlled drug delivery. The third route is to combine the scaffold with the cell therapy, and that offers the ability to know what the conditions are that your cells are going to experience when they go into the patient. So you can actually define the local conditions by creating them with the right type of scaffold. So a couple of examples just of where this has been successful. This is one of the slides where the movies wouldn't play. So the black screen on the left-hand side should be showing you the scaffold. But the key points are on the right-hand images. Here we see three femurs from a mouse model where we've created a critical size defect. So the spaces that you see about a third of the way down are such large defects that they won't spontaneously heal within this animal. And we're looking at a three-dimensional image of these bones after 28 days, and the tissue, as you see, has failed to regrow. On the right-hand side, an example of one of our interventions where we use a scaffold to both encourage new blood vessel formation and new migration of mesenchymal stem cells, and you get complete bridging to occur very rapidly within this model. We're progressing that, on the next slide, towards human therapy using MRC funding. And here we're looking at the problem of hip revision. And you see on this X-ray the problem faced by surgeons where they're putting a second hip implant into a patient. And there's a lack of bone to embed that hip implant in. And what we're looking, if you look in figure A, this is an example of one of our scaffolds, which you create as little pellets, and you can pour into the site created by that metal cage and fill that area with scaffolds, which will then induce new bone formation. And this is where getting the biomechanics of the material is key, because if you can get the biomechanics correct, you can get something that's able to bear the load that goes through that bone and protect the stem cells as they form new bone tissue. So in the last sort of 5 minutes, I just wanted to specifically tell you about the technologies that are being developed by Regenetech. Unfortunately, the movies aren't working again here, but I'm here all day with a laptop, so I can show you if you're interested in it. But the concept here was working with clinicians. We wanted to make this concept of the scaffold easy for them to utilise within the clinic. And one of the key things that an orthopaedic surgeon faces when their patient comes in is a lack of knowledge about the size and the shape of the defect that they're trying to repair. So if someone comes in with after a major road traffic accident, they've lost a large amount of bone, you reset as best you can that bone and perhaps pin it. You then have a large defect which is going to be of a complex shape, and it's going to be difficult to predict it. So what we've created is an injectable material which applies just like a toothpaste. You can inject it into the body, you can press and mould it for about 15 minutes within the operating theatre, and then it will harden, and it will harden so that it has the same strength as spongy bone, and that full hardening process occurs within 24 hours. What you end up with after 24 hours is a material that, if you look through an electron microscope, looks something like this. Lots of particles which are fused together, forming a very elastic shock absorber system, but with lots of porosity. So you'll see lots of space within this structure, which allows mesenchymal stem cells to move in and allows blood vessels to move into the structure. We've done a lot of material science to develop this material. There was no material that could achieve this structure. And again, I'm not going to go into the details during this talk, but happy to discuss afterwards. Most of our technology is based around changing the melting properties of existing polymers that are used in current clinical products, and temporarily making them melt within the body so that you get fusion of particles. And then within 15 minutes, those materials will then spontaneously re-solidify. And that's all around some neat tricks around the use of plasticizers and controlling the behavior of the polymer accurately around about body temperature. And this is the key result, which is that in the wet conditions within the body, with no cross-linking reaction and with no solvents being used, we're able to get spontaneous hardening in these very porous structures. So you'll see this is a study where we're looking at how strong the material is. After 2 hours, we get up to 2 megapascals before the material will fail. That's the lower strength that you see in human spongy bone. And after 24 hours, we're up at around about 4 megapascals, which is about the median value for human cancellous bone within the body. So we've gone on to look at key in vivo models for this. Large defects within large animal models, where you see spontaneous formation within these scaffolds of both new bone and new cartilage. New bone showing in red, new cartilage in blue. And then we went on to look at the drug delivery opportunities. So we're looking at launching a platform technology which can be used in its own right to regenerate new bone tissue. In harder to heal operations, we're looking at partnering with companies with molecules that stimulate more rapid bone formation. And here in these slides, we're looking at the ability of our material to control the release rate of those drugs over periods of 30 or more days, so that our partners with their interesting bone repair molecules can know that those molecules will be concentrated just at the site of regeneration, and that they will have an accurate release of those molecules within the therapeutic window for a sustained period of time. We've gone on to look at complex systems where we're releasing multiple growth factors, and this is a paper that looks at angiogenesis in ischemic muscle models, and looks at, in fact, delivering 2 pharmaceutical products simultaneously in the right concentration gives you a much greater formation of blood vessels, but perhaps more importantly, the quality of those blood vessels is higher if you get the concentration of the 2 factors correct within the scaffold. And finally, just to say that the technology can be used in many other applications, not just within orthopaedics. The concept of having a scaffold that delivers drugs architecture and cells can be applied to many different tissue types. In orthopaedics, we make the material harden so that it's rather like bone tissue. We're also in collaboration with King's College London, Mike Modo and Jack Price, looking at delivering neural stem cells into damaged areas of the brain caused by stroke. And here we're looking at accurately dosing the number of cells. Here the scaffold does not spontaneously harden. It forms into a rather soft gel that has similar elastic properties to the local neural tissue. So just to finish off, I'll leave my many collaborators' names up on the board. I think the field has a huge amount of excitement in terms of opening up therapies that either we just can't solve with current drug treatment, or where biomaterials are not giving us the optimum repair for the entire lifetime of the patient. The problems of getting these therapies through really can't be overstated. They're extremely complex. We're dealing, particularly the regulators, are dealing with the first time they've had to think about how you define a fully functional living cell as a product. But I think the good news is there's lots of opportunities for academics, young companies, to get involved and try and solve some of these problems. Thanks very much.